Morning, everyone. I'd like to get started with the next edition of Science Now, so sit back, relax. We have two really exciting presentations. And you might be looking at the title and wondering what's the connection between the mysterious death of 800 years ago of a young woman and a vegan diet. And I would say absolutely nothing, except that these presentations today will highlight the breadth of research going on in our department and the global impact. So our first speaker is Caitlin Pepperell. She is an assistant professor of medicine in the Division of Infectious Disease. She's an expert on the evolution of human pathogens. And I think somewhat unknowingly, this kind of roped her into a project which I would describe as sort of a very ancient microbial whodunit. So it's generated a lot of media interest, Fox News, ABC News, Smithsonian, and other things. So I hope you'll enjoy that. And then after her presentation, I'm going to talk about our emerging studies using food as precision medicine. So I hope you enjoy.
All right. Oh, good. It's on. So thank you for the introduction and for the opportunity to present this case report. Diagnosing an 800-year-old infection using bacterial fossils. So the story starts in the ancient city of Troy, like Helen of Troy, in Anatolia, which is present-day Turkey. And the reference that I will mostly be discussing is at the bottom of this slide. So I'll start with, like any good case report, with an archaeological analysis of the material. So this was done by Henrika Kiesewetter, who's pictured here. She's an archaeologist and actually is also a physician. So she spends her summers at digs analyzing remains, which sounds like a lot of fun to me. So the cemetery where the remains I'm going to discuss were found dated to the early 13th century and based on archaeological analysis. The people buried there had a pretty tough go of it, evidenced by degenerative disease in their bones and joints, even among the younger individuals. So consistent with hardworking farmers. Short life expectancy. Dr. Kiesewetter did a paleodemographic analysis. So 32 for women, 39 for men. And there was evidence for an impact of pregnancy-related complications on women's lifespan. So a peak of mortality in the childbearing years. And women who survived childbearing actually lived longer. So difficult life, short life. The woman I'm going to discuss was estimated to be, well, first of all, she was estimated to be a woman based on morphological analysis of the bones. And her age at death, estimated age 30 with plus minus 5 years on that. So in prime childbearing years. We did carbon-14 analysis dating of her bones, and that was consistent with the estimated age of the cemetery. So shortly after I started my faculty position here at Madison, I received this gift, which are these nodules. So this is actually a picture of the nodules on my desk. In the little baggies that they came in. I got this gift from Dr. Aylward, who's pictured here. He's a professor of classics here at UW-Madison and has been involved in excavations at Troy for a very long time and then took over the project. He's close collaborators with Dr. Calcified, and she made him aware of these nodules. So I'll tell you a little bit more about them. You can see them as well. They're about the size of strawberries, so supermarket strawberries, not wild strawberries. They're chalky white, and Dr. Calcified found them at the base of the ribs of this 30-year-old woman. And her initial, and I think very good thought, is that these might represent calcified granulomas from tuberculosis. And this is a reasonable thought because tuberculosis lesions do calcify readily, and certainly it's been an important disease, would have been an important disease in that time and place. So this was her initial thought. And when I talk about the nodules and Dr. Calcified's discovery of them, I do like to emphasize how careful and perceptive she was in identifying these small, chalky lesions. As I'm sure you can imagine, it's very difficult to separate material like this from the dirt and rocks that are all over the remains. So big credit has to go to her for her careful work and perceptiveness in recognizing the potential significance of this material. So William came into possession of the nodules, and he found me, and he was familiar with my work in the evolutionary genomics of Mycobacterium tuberculosis, and he thought that I would be a good person to investigate this further. And we hope to retrieve some DNA from an ancient pathogen, which is really helpful in understanding pathogen evolution. So it took me no time at all to say yes to this project. And my first task at that point was to find an expert in ancient DNA analysis. So someone who can handle the material, extract it. It has to be extracted in a specific way, and help with the initial processing and analysis of the data. I contacted Hendrik Poinar, who is a renowned ancient DNA specialist at McMaster University, and it took him about 1 second to say yes to analyzing this material from the fabled city of Troy and try to get to the bottom of this infection. The usual scenario with this kind of work is that you start with what's called shotgun genomics. So essentially you just sequence everything that's there. And you hope that your target, so in this case we were thinking MTB, you hope that you'll find it at an abundance of something like 1%. And this will be a lead finding that will take you down the path during which you enrich for your target material. So that's what we went into it expecting. We thought it would be TB. We thought if we're lucky, we'll get 1% of the material, the sequence data mapping to MTB. So what we did get was a total surprise. It was absolutely not TB. The work, the initial work putting together the genome was done by Alison DeVoe, who's pictured here, and was a student at the time in Hendrik's lab. So we got extraordinary amounts of endogenous ancient DNA. DNA has particular physical and chemical signatures of degradation over time, and so you can use these signatures to identify the sequences that are bona fide ancient DNA. So as opposed to the sort of 1% that we were hoping for, a third to two-thirds of the DNA from the nodules was from this bug, Staph saprophyticus. This is the first ancient staph data that has ever been acquired. So huge proportion of staph, good proportion of ancient endogenous human DNA. We checked the mitochondrial haplotype of the human DNA against the bone, and the nodule and the bone were a match, and confirmed a female origin. And then, in addition to the Staph saprophyticus, also a significant component of Gardnerella vaginalis. The quality of the Staph saprophyticus DNA sequencing data was such that Allison was able to reconstruct de novo a complete genome at an average of 300-fold coverage. So by far the most complete ancient bacterial genome to date. So who are these critters in these mystery nodules? Staph saprophyticus is a coagulase negative staph. It's a funny one in that we don't usually think of staph associated with urinary tract infections, but this one is actually quite an important cause of urinary tract infections, and is in fact second to E. coli as a cause of UTI in women of reproductive age. It can be found also as a member of the GI and GU microbiome, usually as a transient member. Gardnerella vaginalis, as the name implies, is part of the vaginal microbiome. It's also associated with a dysbiosis called bacterial vaginosis, and occasionally with invasive infection even outside of the genitourinary tract. So definitely not tuberculosis, and these bugs locate us in the genitourinary tract. So a sort of plausible alternative diagnosis at this point was that these calcified lesions were renal stones, and that we know that Staph saprophyticus and other urinary pathogens can grow inside of bladder stones, and that there's an interplay between infection and stone disease. So that made sense. And then Gardnerella vaginalis is not a urinary pathogen, but it's quite commonly found in the urine and in urine cultures. So definitely a plausible alternative diagnosis. So to help sort this out, we did elemental analysis using XRD. And to make a long story short, this was not consistent with stone disease. So the typical components of bladder stones, particularly infected bladder stones, struvite and calcium oxalate, were not present. And instead we had these other elements, primarily hydroxylapatite and whitlockite, which are components of ectopic. They're found as part of ectopic mineralization. So inflammation and other causes of ectopic mineralization are associated with these particular elements. So we proceeded further to characterize the tissue using scanning electron microscopy. I can see Janelle has a smile on her face. She loves to look at SEMs. So the SEM was quite amazing. What we saw were what we termed, and a term we coined for the paper, ghost cells. So these are mineralized material in the size and shape of staphylococci. So coxoid-shaped material that you can see over here. So clusters, essentially clusters of staphylococci. Here are what are we call trace fossils. So these are imprints in the mineral matrix in the size and shape of the staphylococci. And then over here you can see this stringy stuff, which is similar in appearance to EPS, which is a component of biofilms. We also found material in the size and shape of inflammatory cells, so what look like mineral shells around neutrophils at the site of infection. I showed these SEMs at a talk I gave at the University of Illinois, and one of the people in the audience studies Staphylococcus aureus, and he came up to me after the talk and said, those look just like SEMs of Staph aureus biofilms. And he was amazed that this ancient material had preserved not only the shapes of the individual cells, but the architecture of the tissue. The term ghost cells, or one of the concepts attached to this, is that we think that these shells, these cellular shells, had a really important role in preserving the endogenous DNA. So in phraseology that I'm sure will haunt me for the rest of my career, I talked about tiny suitcases of DNA to a reporter when I was describing this, that transported the DNA over this 800-year time span, protected it from contamination and physical and chemical assaults. So this is why we think we got such an amazing amount of endogenous DNA from this material. All right, so let's get back to the medical mystery. So the nodule is pictured up here, or one of the nodules is pictured up here. Based on the amount of bacterial and human DNA, we're looking at an active infection, such as an abscess. We know that we're in the genitourinary tract. It's a mixed infection. Gardnerella vaginalis is important. It's an infection that's associated with ectopic calcification. And then the final sort of smoking gun was the discovery of a minority male component to the tissue. So the bones and the nodule were sexed as female, but there was a minority component of ancient Y chromosome. Putting all of this together, I settled on a diagnosis of chorioamnionitis. And the underlying rationale for this is the organisms, the fact that it was a mixed infection. Staph saprophyticus has been associated with chorioamnionitis. Gardnerella vaginalis is typically identified in cases. The pattern of calcification, so the placenta calcifies readily as a result of calcium flux between mother and fetus. And in cases of infection, particularly chorioamnionitis when it involves the umbilical cord, which is called funicitis, the calcification can be so pronounced that you can't actually clamp the umbilical cord. It's just completely hardened. So it seemed like a good setting in which we would see this rigorous ectopic calcification. In addition, of course, we had the archaeological setting and the knowledge that in this region, as in others historically and to this day, pregnancy-related complications and infections are an important cause of mortality among women. And then, of course, we had this Y chromosome data, which really nailed it for me. And one of the things that I learned about chorioamnionitis as I was researching this project is that the inflammatory response comes from both mother and fetus. So I hypothesized that this Y chromosome material was from a male fetus and from the involvement of his inflammatory cells in the infected tissue. So that's the medical mystery part of the story. At this point, I take off my doctor hat and put on the scientist hat, and what can we learn about bacterial evolution? So the work I'm going to present now was done by Tatum Mortimer, who's a graduate student in my lab defending her thesis next week. And she, at the time that we started the project, there was very little genomic data from Staph saprophyticus. So I went on a hunt for people to give me isolates from around the world, collected them, and did whole genome sequence analysis. So as you can see from the little icons on this tree, Staph saprophyticus is actually associated with all kinds of different material, including you can find it free living in the environment, in ice cream and sausage and other foods, in cows and other animals. The Troy isolate was positioned over here in a really interesting place. So up here we had an isolate that was associated with bovine mastitis. And then here we have a concentration of human urinary isolates. So these are all human urinary isolates with the exception of this spillover into a dog. So we identified this lineage that seemed to have a particular propensity to cause human UTI amidst a potpourri of other niches that this bug is adept at inhabiting. So Tatum and I were curious about whether we could find any signatures of specific adaptation to the human urinary pathogenic niche. So Tatum, we looked in the population genetic data for just such a signature, and we found evidence of a selective sweep, which is a population genetic term, but it has a really intuitive meaning. And essentially what it means is that you have an advantageous mutation arise. It increases in frequency in the population very quickly, and it leaves a particular signature in the genetics of that population. So we identified a selective sweep in this human-adapted lineage, and this is actually the first selective sweep identified in bacteria. We localized the advantageous mutation to an adhesin called AAS, and adhesins are really important for urinary pathogens. Rod Welch, who's my MMI department chair, likens the job of a urinary pathogen to grabbing onto slippery rocks while a waterfall cascades over you. So they really need their hooks. And AAS is known to be one of these important hooks. The mutation occurred in the fibronectin binding region of AAS, and we did follow-up work with Dean Mosher. Douglas Annis from the Mosher lab is pictured here. He did the work and showed that the mutation, the novel mutation, altered binding to human fibronectin. So it increased the avidity of the bacterial protein for this human protein. So this provides a plausible mechanism by which this particular lineage would be adaptive and particularly well-suited to the human urinary pathogenic niche. Tatum did some demographic reconstructions and estimated that movement of Staph saprophyticus into this niche was associated with a large-scale expansion. So in summary, we used archaeological, ancient DNA, metagenomic, elemental, and SEM data to diagnose and characterize the first pregnancy-related infection in the archaeological record. This led to the identification of a UTI-adapted lineage of this UTI pathogen. I imagine that most of you will not be diagnosing diseases from the archaeological record, and maybe you're wondering what this has to do with clinical medicine now. And what the relevance of it, I think, or one of the avenues of clinical relevance is in this idea of using shotgun sequencing diagnostics. Just sequencing everything in a material to get at a diagnosis. Pretty much every time I'm on clinical service, I wish I had these technologies at my fingertips for difficult to diagnose cases. And this is very, an active area of investigation. Of course, like everything else, the devil is in the details. In this case, we have things like the signatures of ancient DNA to guide us to our target. When we're in the clinic and it's all modern DNA, it's harder to sort the wheat from the chaff. And in a clinical situation like this woman's, I think, if she was a living patient, we would have been able to culture those organisms probably quite readily. We actually got the staph saprophyticus from her ulna, indicating that she was bacteremic. So the situations where we really want shotgun diagnostics are the situations in which the data are most difficult to analyze, where the organisms are found at low abundance and/or they're not usually associated with this particular clinical syndrome. So promising, but stay tuned. And I'll end with my acknowledgements. My lab, particularly Tatum, and collaborators on and off campus, and my funders. And thank you so much for listening. Yes? Sorry, what's that? Yeah, so the question was about the demographic reconstruction and how we did that. So there are methods based on mathematical models of sequence evolution of reconstructing the evolutionary history. So you start with data from an extant population, and then based on models of how population genetics change over time, you reconstruct how you got to that point in time.
So I might have missed it, but I think you mostly talked about the staph. Was there enough Archeological DNA to place it in a unique evolutionary niche, or?
Yeah. So I didn't talk about the Gardnerella just out of time, but it's an absolutely fascinating bug. Yes, we did place it, and basically the Gardnerella vaginalis of 800 years ago is totally indistinguishable from the bugs that are around today. And we think that this is probably because of rampant lateral gene transfer, which tends to preserve diversity over time. I think it probably played a role, yeah. It's a tricky bug. Yeah, I'll just leave it at that, and we can talk some more offline, because I could talk for hours about it. Thank you.
So let's talk about food next. So I have nothing to disclose, except that I hope that after my talk, you'll never look at a crown of broccoli in quite the same way. So can we think of food as precision medicine? And the idea here is, can we use food to reprogram our genes? And I'm sure you're thinking, really? So we've heard you are what you eat. This is a saying that goes way back, and it was taken to its logical extreme by this Italian Renaissance painter Arcimboldo. And this is the Roman god of seasons. So we know that food influences our susceptibility to disease. But what I'm talking about is, can we use food as medicine? Can we unlock the molecules in our food and treat diseases like cancer and diabetes? So my lab focuses on triple-negative breast cancer. These are cancers that lack the estrogen receptor, progesterone receptor, and HER2. They account for about 10 to 20% of all breast cancers. They're more prevalent in African American women and Hispanic women. And they're associated with poor outcomes, mostly because they metastasize to lung and brain. And unfortunately, we don't have targeted agents. So this is an area of great unmet need. So we know from epidemiologic studies that vegans have the lower incidence of certain types of cancer. Vegan diets are low in methionine, and this is an essential nutrient that plays a role in lots of different biochemical reactions. We also know that tumors require methionine for their growth, but clinical trials of low methionine diets in cancer have been mostly disappointing. So what we reasoned is, if we could uncover molecular mechanisms, we could make methionine restriction work better as a cancer therapy. And that's really the goal of what we're working on. So let's talk a little bit about methionine. So methionine regulates gene expression through its conversion to S-adenosylmethionine, or what I'll call SAMe. You can actually buy this in GNC, and hopefully after my talk today, you won't do that. But so SAMe donates methyl groups to DNA directly and to histones and alters gene expression. And it turns out that when we expose tumor cells to low methionine, they activate these stress response genes that allow the tumor cells to survive that low methionine stress. And so what we reasoned was that if we targeted those stress response genes, we could eradicate the tumor cells. And we call this metabolic priming. It's sort of a 1-2 punch. And one of the first targets we identified when we methionine restrict tumor cells is a receptor present on the surface of tumor cells called trail receptor. And this receptor actually is engaged by immune cells, like natural killer cells, and kills those tumor cells. So it's a known cell death regulating receptor. And somewhat unimaginatively, there are two trail receptors named trail receptor 1 and trail receptor 2. And so what we did first was take tumor cells and methionine restrict them. And what happens when you do that is both trail receptors' expression goes up. And if we take tumor cells and expose them to low methionine or to a trail receptor, you can see there's fewer, there's cell death because there's fewer healthy blue cells here. But if we combine them, we virtually eradicate all the tumor cells. And we can study this in the mouse using fluorescently labeled human triple-negative breast cancer cells, and then treat the mice either with a low methionine diet or a trail receptor antibody or the combination. And we're looking here at the lungs removed from these mice, and you can see low methionine diet has some effect. Trail receptor has some effect, but the combination, again, is much more powerful at reducing lung metastases. So I've made this cartoon to kind of summarize the metabolic priming strategy that we're using. So methionine stress activates trail receptor gene expression, and this leads to increased trail receptors on the surface of the tumor cells, and now it primes them to respond to our trail receptor drug, which then kills the tumor cell. Now, that actually all worked. The other thing that I think is really distinguishing about this metabolic approach to cancer is that methionine restriction kills these so-called cancer stem cells shown here. And these cancer stem cells are resistant to traditional cytotoxic chemotherapy. So this is a huge advantage. And it's thought that the persistence of these cancer stem cells plays a key role in treatment resistance and metastasis because they're not eliminated by cytotoxic therapy. And it turns out that we can actually rescue cancer stem cells that are exposed to methionine restriction by SAMe. So it's really not methionine that they require, it's SAMe, presumably through its changing the epigenetic program of these cancer stem cells. So interestingly, what happens when you methionine restrict tumor cells is the enzyme that converts methionine to SAMe gets massively induced. And that's shown here. This is cell line data, but we see the same thing in tumors in mice. So this enzyme gets massively induced, and presumably that's to maintain homeostatic levels of S-adenosine methionine or SAMe. So we postulated that if methionine restriction basically induces this enzyme to preserve SAMe levels, what would happen if we use an inhibitor of this enzyme? And cycloleucine is a non-metabolizable amino acid homologue that inhibits MAT2A. So if this is critical, we would predict that this combination of low methionine and this inhibitor would, again, result in robust tumor cell killing. And that's exactly what we saw. So here again, we're looking at our cancer stem cells. And if we treat them with low methionine that inhibits their growth, if we treat them with this MAT2A inhibitor, cycloleucine, it also inhibits their growth. But if we combine the two agents, we virtually eradicate these cancer stem cells. Again, another example of metabolic priming. So that's kind of some of the work that we've done in cancer. Can we use this approach for diseases like obesity or diabetes? And this is work that I've done with Dudley Lamming. I think Dudley's here somewhere in the back. What we asked was we took female mice and we put them either on a control diet or a Wisconsin high-fat diet. And after 12 weeks of that Wisconsin diet, we took the mice and split them into two groups. One group, and they both stayed on the high-fat diet. One group got the high-fat diet with no methionine, and the other group got the high-fat diet with a normal complex of amino acids. And what we saw was really striking. So the green is the high-fat, no methionine. So these mice eat more, yet they lose weight rapidly. They become, over 5 weeks, they become leaner than the mice on the control diet. So this is quite dramatic. And virtually all of the weight loss is due to loss in visceral fat. So they're losing visceral fat, very little loss in lean mass. And to make things even more interesting, these mice on the high-fat diet and no methionine exercise less. And these changes are accompanied by improvement in fasting blood glucose, lower fasting blood glucose, and improved glucose tolerance. So what I'm telling you basically is these mice on this high-fat diet with no methionine, they're eating more, exercising less, and they're losing massive amounts of visceral fat. So if Dudley and I had a pill that could do that, we'd never have to write another grant in our lives. So we're working on that, but we don't have it yet. And of course, we want to go beyond descriptive. So we want to understand, how does that fat all disappear? And so one of the tools that we use is transcriptomics. And so we can look at the messenger RNA in virtually all the tissues in response to methionine restriction. And this is just data from liver tissue where we're doing a pathway analysis. And basically, a number of metabolic pathways are either decreased or increased in the liver in response to methionine restriction. And why is this interesting? Well, these are going to provide us with some essentially roadmap for drugs that we can use to perhaps mimic the effects of methionine restriction. Again, we're trying to find ways to avoid writing grants for the rest of our lives. So what's really exciting, I think, about this work is, even though I've had a lab almost for 20 years, I've actually never taken a discovery from my laboratory directly into the clinic. And that's changed this year. So we actually have funding for 2 different clinical trials in triple-negative breast cancer. And this is work that I'm doing together with Ruth Reagan, who's also here. And thank you for coming, Ruth. And so what are these 2 trials? Well, one is a trial in women with newly diagnosed triple-negative breast cancer. So here the idea is, before their surgery, we're going to put them on a low methionine diet and look at what effect that has on their tumors and on their metabolic health. How are we going to do this? Well, it turns out that if I just give you a list of low methionine foods, maybe everyone in this audience would be incredibly diligent in following that list, but maybe at best we might get a 40% reduction in blood methionine levels. We want to do better than that. So what we're doing is providing them with a medical food that has 19 of the 20 amino acids. It's missing methionine. And then we also give them a list of low methionine fruits and vegetables. The second study that was funded is that we're going to do a statewide phase 2 trial in metastatic triple negative. We're going to use a new trail drug, ONC201, either by itself, and the second arm will be this drug in women who are on a low methionine diet. And we're going to look at their treatment response. And this is just to show why we're using this particular drug, ONC201. It's a new drug that has certain advantages. It's orally available. Many of the other agents that I talked to you are monoclonal antibodies that have to be given parenterally. So that's a big advantage. It's been shown to be safe. It also has effects on cancer stem cells. And really important for this subset of patients who have high rates of CNS metastasis, this drug crosses the blood-brain barrier. I show you here basically some preclinical data showing that the combination of low methionine in this drug, again, virtually eradicates the tumor cells. The other cool thing about these clinical trials is that they're incorporating transformative technologies, transformative local technologies. So we're going to do a so-called liquid biopsy to look at these rare circulating tumor cells using a device co-developed by Josh Lang, a lab on a chip. So I don't know if Josh is here, but he's built these really tiny labs on chips that use essentially antibodies linked to magnetic beads to sort of pull down and capture these circulating tumor cells. And then one can then query both their message levels and protein expression all on the same chip. It sounds like science fiction, but it really works, and it's very exciting. And of course, this will enable us to look at, the effects of our interventions in that metastatic study where we don't have access to tissue, but we can look at changes in the circulating tumor cells. And the other transformative technology is pioneered by Josh Kuhn, who's in biomolecular chemistry, and he's built this high-throughput mass spectrometry assay to look at not only methionine, but other metabolites in the methionine pathway. And we can do this, of course, in our patients in the clinical trial to show that our low methionine diet is not only lowering plasma levels of methionine and its metabolites, but also tissue levels. Particularly, we're interested in the tumor expression. So this, definitely takes a team, and the team has members that are clinicians, members that are basic scientists, members that are community advocates, and has collectively a lot of expertise. So, as I mentioned, Ruth and I are leading this study. We have expertise in clinical trials, pathology, nutrition, medical imaging. We're using MRI. Scott Reeder has developed some innovative platforms for using MRI to quantitate visceral fat in various regions. It's really important that we have cancer survivors and advocates because we're going to have to recruit a lot of patients to these trials. It's always dangerous to show Oprah a little early, but it's foreshadowing. Okay. So we have expertise in metabolism, metabolomics, as I mentioned, and circulating tumor cells. So there's we've brought together a very multidisciplinary and talented team. So Dr. Page always says we're not very good about bragging about what we're doing. So These slides were included for him, but of course he's not here today, so you'll have to tell him that there was a lot of bragging going on. And so this metabolic, what we called metabolic priming, Oprah called the vegan solution, and it was listed as one of the 3 breakthroughs in breast cancer in 2015. And if that's not surprising enough, Ruth and I were actually on ESPN during Jimmy V Week, so we'll have to show this little clip. I compared Ruth to, I called her, in one clip they didn't take, the Aaron Rodgers of breast cancer clinical trials. But this is what they took. If I can figure out how to get it to go, let's see. There we go. Maybe. Da, 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 da. Stop. I might have to ask Clint.
I'm Ruth O'Regan. I'm a breast medical oncologist at University of Wisconsin.
And I'm Dr. Vincent Cryns, and I'm chief of the endocrine division at the University of Wisconsin. The best part about my job is that our research is giving hope to patients with metastatic breast cancer. And I'm very hopeful that the kinds of research we're doing are going to make a difference in patients' lives. And if the Cubs can win the World Series after 108 years of trying, anything is possible.
So the most rewarding part of my job is taking care of patients with breast cancer, particularly when we find a new therapy that actually helps them.
All right, an assist to Clint on that. He was kind enough to provide that video for them. And, of course, to the Chicago Cubs, who seem to be not doing so well this year. So in summary, then, I hope that I've convinced you that we can use a low methionine diet to fundamentally and globally change gene expression. We're using this as a new therapeutic paradigm where we stress the tumor cells with low methionine, and then we target those stress response pathways. And it's this sort of 1-2 metabolic priming approach that I think is going to be really powerful. I've also shown you some of our early studies where we're using a low methionine diet to fight obesity-related conditions like diabetes. And of course, the thing that I'm most excited about is that we are now actively translating some of these discoveries into clinical trials. And I'm not sure they're going to work, but we're hopefully going to find out in the next couple of years. And thank you for your attention. So I'm ready for a couple of questions in the back.
A number of years ago, an investigator, did a large randomized study with an extremely low-fat diet. And the original hypothesis was that it was going to affect ER-positive breast cancer. But in point of fact, what they demonstrated was a marked improvement in survival with a rate to hazard ratio in triple-negative breast cancer. A subsequent paper in the journal, JNCI journal, attributed it to insulin receptor. And I was wondering how those data integrated with what you were talking about.
All right, I'll try to summarize that. Some studies on a low-fat diet. I'm not aware of these studies, by the way, so I'm going to correct. I'll hopefully respond, copy what you've said. Low-fat diet had some effects in triple-negative breast cancer that subsequent studies perhaps showed that the insulin receptor might be involved. I can't comment on that directly. What I do know is that we've been talking, most of the limited slides that I've shown have been looking at the direct effects on the tumor of this intervention. But clearly there are metabolic effects, and there's a loss in visceral fat. So my guess is that what we'll see in vivo in people and in mice is that it's a combination of direct effects on the tumor as well as systemic effects, improved insulin sensitivity, lowering of for instance, IGF-1 levels, lowering of insulin levels. But I don't have data for that specifically, but it's something that we're looking into. Yes, Yoram.
What is the intervention, the planned intervention in metastatic cancer patients? They would be worried about compliance. If you plan to do it for a year or two, maybe it will be.
Yeah, so So the question is, how long is the intervention? And I think that's one of the exciting things. What we're talking about in this metabolic priming approach is a very brief intervention. Activate the stress response, and then combine that with a targeted cancer drug. So, for instance, in this clinical trial, the patients are 5 days, and then they cycle off. So the dietary portion is 5 days, then they cycle off, they recover, and then they go back on. And they'll stay on until they progress, and that's how the trial's written. So it's not a sustained methionine restriction over time, although that's been done by other individuals. It also in metastatic, in patients with metastatic melanoma, were put on a similar diet for up to 39 weeks. Now, the results were disappointing, but we would say that's because they didn't have the molecular insights that we have, that the effects of just the dietary intervention are quite modest. You combine it with the priming part. Any other questions? Yes? Oh. Sorry, no, that's fine. Go. Why don't you start? So the question is whether I'm liberally paraphrasing your question. So hopefully I catch the spirit. Has methionine restriction been implicated in longevity? Is that? Yes, and that's not our work. Although my collaborator Dudley Lamming has an interest in aging, but there are rodent studies showing that methionine restriction definitely increases longevity. In much the same way that caloric restriction and protein restriction. And one of the challenges in the field is to figure out how these different interventions, whether they have similar common pathways, partly overlapping. Of course, in my methionine-centric world, I would say that perhaps caloric restriction is going to lead to methionine restriction. Protein restriction is going to lead to methionine restriction. So I'm not saying that methionine is everything, but probably some of the contribution from these other types of diets may be to limited availability of methionine in them as well. And you had a question in the back. So the question is, do we think there's any role for this in breast cancer prevention? So, of course, I would say yes. But the issue there would be talking about long term. So what we can't do, which is what we're doing currently, is trying to get methionine levels to plummet precipitously, which would not be tolerated over a longer period of time. That question about whether it can be preventive, and we are looking at that in mouse models, would require more modest reductions in methionine over longer periods of time. And whether that works, we don't know yet, but that's something we're actively looking at. Yes?
Has any thought about how this might interact with African Americans being more likely to get triple-negative?
That's a really great question. So let me repeat it. Give you credit for that great question, Chris. The question is, could the higher incidence of triple-negative breast cancer in African American and Hispanics have something to do with their methionine intake? And I have absolutely no data to support that one way or another, but I wouldn't be surprised. If we're talking about methionine playing this pro-tumorigenic effect, we know that people's dietary intake of methionine varies widely. There's a little bit of data in normal human subjects. Fasting levels of methionine in the blood in normal human subjects ranges anywhere from 3 to 30 micromolar. That's a huge variability. And it does seem to reflect with dietary history and BMI. So people who are overweight and eat a lot of meat are going to have higher methionine levels. And to what extent that's contributing to their enhanced incidence of cancer is an open question, but I think it's a very, very legitimate and interesting question. Yes? Len? So that's a great question. Len's question is, what happens to the trail receptor in non-cancer cells? So interestingly, when we look at the expression of trail receptors in non-transformed cells and in normal tissues, the cell surface expression doesn't seem to go up. We don't have an explanation for that, but it would provide some explanation for the observation by us and others that this metabolic strategy seems to be to preferentially target tumor cells as opposed to normal cells. And again, I don't know why the receptors don't get upregulated, but they don't seem to do so on the cell surface. All right, thank you for your attention.